kick off. I was waiting for the co-chair, but apparently the co-chair's not here. So are you happy to just get going?
Sure.
What I'll do is 5 minutes, 10 minutes. And then we've got 5 minutes for questions as well anyway. Okay. So if you're happy to go, we'll get started, yeah?
Okay, that works.
Okay, good. Afternoon, everybody. Welcome, I think, to the final session of the day. We're a little bit late getting started. I'm going to chair the session. My name's Gary Stevens. I'm from the University of Reading. So without any further ado, I'll introduce the first speaker. We've got 10 minutes for talks and then 5 minutes for questions. If you do have any questions, it'd be great if you could go up to the microphone, just introduce yourself and ask the question. So our first talker is Daniel Morgan, and he's going to tell us about mutant mice expressing an internalization-resistant form of CB1, cannabinoid 1 receptor display enhanced cannabinoid tolerance. Sorry, Dan, I hope you make a better job than I do of than your title.
Introduction. So I have to be honest with everybody. So my flight actually arrived at 2 a.m. last night. So yay, like I'm here. My luggage did not. So hopefully I don't smell too bad. You guys will have to let me know. And I'm going to try to be coherent despite the relative lack of sleep over the last few days. So nonetheless, I'd like to thank the program committee for giving me the opportunity to talk about the work currently going on in my lab, looking at mechanisms of cannabinoid tolerance. So chronic pain is a widespread, unmet medical need with a worldwide prevalence of about 6 to 7%. Currently available treatments for chronic pain really only work in about half of chronic pain patients. So really one of the reasons why we're interested in studying cannabinoids is their potential use as agents to treat chronic pain. So in 2016, the US National Academy of Science organized a committee to assess the therapeutic benefits of cannabinoids in a wide range of different clinical conditions. Some of those conclusions are shown here, including evidence or their conclusion that there was strong or substantial evidence that cannabinoids were effective for the treatment of chronic pain in adult patients. However, one limitation to the long-term use of cannabinoids for the treatment of chronic pain, or for any treatment or recreational use for that matter, is that with heavy use, there's the potential for tolerance to develop. So in my lab, we're really interested in understanding those mechanisms of tolerance, and essentially we're interested in understanding what's going on with this guy using mutant mice as our model organism in the laboratory. So it's well known that 2 serines in the cannabinoid receptor 1 are required for the classic mechanism of receptor desensitization. So this is work that was done by Ken Mackey's lab about 15 years ago. And this really shows a seminal piece of that data here. So you can see that exposure to a cannabinoid agonist, this is in transfected cells, that you get this robust activation of ERK1 and 2 signaling. In cells that express wild-type CB1, there's this very rapid desensitization of that signaling response. In contrast, in cells expressing this S426, S430A mutant form of CB1, that desensitization is blocked. So when I became a postdoc with Ken many moons ago, one of the things that we were interested in doing was making knock-in mutant mice, where we're essentially substituting these 2 putative GRK phosphorylation sites for non-phosphorylatable alanine residues. So that data is shown here. It's published data. It's been a while ago, so I don't want to belabor it. But what we found is that by blocking this classic mechanism of desensitization in mice, that we see a modest 1 to 2 day delay in the development of tolerance. And this is looking at once daily injections of 30 milligrams per kilogram of THC. So what we're working on now, and really the point of the talk, is looking at this second cluster of putative GRK phosphorylation sites. So it's known that there's another cluster of 6 residues. It's located more distal in the CB1 terminal in the tail. So we're interested, based on this data that, again, it was done by Ken Mackey about 15 years ago. And what they're showing is that if you mutate these 6 additional residues, that you get this block in internalization with a really very modest effect on desensitization. So this additional site seems to be fairly selective in terms of impacting internalization versus desensitization. So that really provided the critical rationale, at least in my mind, for producing 6-point mutant mice. So again, what we're doing is we're making knock-in animals that express these alanine substitutions at these putative GRK phosphorylation sites. And really the idea behind this was to test the hypothesis that if we block internalization of CB1, that's likely to enhance tolerance for strongly internalizing cannabinoid agonists. So the rationale for that hypothesis is really rooted in the complicated fate of internalized GPCRs. So it's known that in order for desensitized GPCRs to be resensitized so they can signal again, that they need to be internalized, at least for this process to be efficient. There are other mechanisms, but you need internalization, action of phosphatases, which then dephosphorylate the receptor, and then at that point it's able to be resensitized or recycled to the membrane where it can signal again, or it can be sent to the lysosome and degraded. So this idea really gave rise, like in the late 1990s, to this rave hypothesis that tolerance or desensitization for agonists might be different for strongly versus weakly internalizing compounds. So indeed, when we look at tolerance for cannabinoids, and this is just an experiment in wild-type mice, in this case, we're looking at tolerance for THC, which is a weakly internalizing agonist, versus CP55, which is a strongly internalizing agonist. You can see that for THC that we see tolerance very rapidly, so within 2 to 3 days. In contrast, when we look at tolerance for this strongly internalizing agonist, it's much slower and gradual. So we've produced these 6-point mutant mice. They've been in the lab for about 6 months at this point. One of the first things that we've done is we've performed dose response curve analyses looking at CP55. So these are cumulative dose response curves. And what we see when we look at anti-nociception, either using the hot plate or the tail flick test, or we look at hypothermia, we get a genotype effect. And what that indicates is that the response in these 6-point mutants is reduced relative to wild-type animals. When we look at tolerance to CP55, so this is going to be once daily injections of 0.6 milligrams per kilogram of CP55 over a course of 10 days, we see a genotype effect indicating that the response in the mutants and heterozygotes is lower than wild type. We also see a dose effect or a day effect, which means that over time there's a reduction in response, right? So there's tolerance developing. It's hard for me to say at this point whether the tolerance is different in the 6-point mutants versus wild-type animals. And the reason for that is the analysis that we've done. So this is going to be a two-way repeated measures ANOVA. So we don't get a genotype by day interaction effect. So at this point, I think it's too early to say that tolerance is impacted one way or the other. So what's known is that if you disrupt beta-arrestin recruitment to GPCRs, that this tends to have a pretty dramatic effect in terms of the magnitude and duration of an acute injection of either an opioid agonist, such as morphine, or a cannabinoid agonist, such as THC. So you can see here, this is work done by Laura Bond almost 20 years ago, that in beta-arrestin 2 knockout mice, where beta-arrestin's been deleted, that there's this enhancement in morphine response in the tail flick test. In contrast, when we look at THC in our S426-430A mutant mice, we actually see a really similar response. So we see this enhanced hypothermic response to THC. So one of the things that we were interested in our 6-point mutants was whether there was a change in the duration of effect in these 6-point animals. And in fact, that's where we see the most dramatic impact of this mutation. So in this case, we're looking at a bolus injection of 0.6 mg per kg of CP55, and we're looking at the response over 5 hours. So what you can see is that for both heterozygotes as well as mutant animals, that there's really this profound and robust decrease in response and duration of response for both the tail flick for the antinociceptive effects as well as the hypothermic effects of CP55. So over about the last month or so, we've started to characterize THC response. So this is obviously something that we're going to be really interested in. So again, this is looking at cumulative dose response curves for both the antinociceptive effects of THC as well as the hypothermic effects of THC. So in this case, we're actually using heterozygote animals. So we actually don't have very many of these mice at this point. So we're just trying to get a basic idea of what's happening. When we look at antinociception, there's really no difference in the dose response curve between mutants or between heterozygotes and wild types. In contrast, when we look at hypothermia, there's this slight reduction in response. So that suggests that the hypothermic effect, but not the antinociceptive effect, seems to be impacted, at least for THC. So when we look at tolerance for THC, so again, we see a genotype effect for the antinociceptive response to THC. We don't really see any impact on the hypothermic effects of THC. So this is a genotype and also a day effect, but similar to CP55, we're not able to see with this low number of animals that we have at this point an interaction effect. So it's hard to know whether there's an impact on tolerance or it's just an overall decrease in response in these mutant mice. So finally, so we've also looked at the duration of THC effects. So this experiment was done similar to what we've done for CP55. So the animals get a bolus injection of 30 mg per kilogram of THC. The experiment's done in both wild type and heterozygote animals. We really don't see any difference in the duration of effect for the tail flick, for the antinociceptive effects. We do see a decrease in the response, THC-induced hypothermia. So with that, I just wanted to briefly summarize what I've showed you. So when we look at dose-response curve effects in these mutant animals, so we see a reduced response to all effects of CP55, 940. We see an effect in the hypothermic response to THC, so this impact seems to be both agonist and response specific. When we look at tolerance, it's not totally clear what's happening at this point. We really need a larger groups of animals to be able to say for sure, but there's a possible increase in tolerance for the antinociceptive effects of both CP55 and THC, and that effect seems to be response specific, so we don't see any impact in hypothermic tolerance. Really where we see the biggest effect so far is in terms of this duration of effect. So for both the antinociceptive and hypothermic effects of CP55, we see this decreased response. And for THC, we see a decrease in the hypothermic effects, but not an effect on antinociception. So with that, I just want to thank members of my lab that have really been amazing over the last few months, like churning out this data. So we haven't had these mice for very long. Collaborators at Texas Tech and Indiana University, and also generous funding from NIH that supported this work. So with that, I'm happy to take any questions.
Thanks, Daniel. Any questions for Daniel? Yeah, can you just go to the microphone?
All right, I'll shout.
Okay, go on.
Basically, your first sentence I'd like to take issue. Actually, 50% of people get benefit from medications for chronic pain at the moment. That's a very dangerous statement.
Okay.
That's a comment. Yeah, I mean, I guess opioids and cannabinoids, are we looking at the same issue here? I mean, you looked at tolerance, but what about dependence?
Yeah, so
Like working with THC, for example. Not suggesting THC is going to be used.
yeah, so some of those experiments are really difficult to do in mice. So we can look at precipitated withdrawal. So that's something that we've done for the S426-430A mutant mice. And it actually precipitated withdrawal is enhanced in those mice. So we might predict an opposite effect in these. But to really do voluntary self-administration assays, that's something that's been very difficult in the field to get mice to do that. So they actually don't like to self-administer cannabinoids. So, but it's a good question.
I'll ask one. Do you have any idea of the kind of opioid sparing effects in your model then? Is that something you're looking at?
So we haven't looked at that in these mice. We did look at these S426, 430 mice that were published a few years ago. So we did look at morphine tolerance and precipitated withdrawal from morphine in those animals. And my memory, there wasn't really a difference. So it doesn't seem to affect morphine response.
Okay.
So.
And you intimated this at the end, how agonist dependent do you think these effects are? So THC is partial agonist and CP55 is full, and
yeah.
You're going to look at Win55 as well, yeah?
Yeah, so that's actually next on the list, actually. So it's a good point. Yeah, I mean, that's sort of been our bread and butter over the years, was to look at these agonist-specific effects. And indeed, I mean, I don't know what's going to happen in these mice. Actually, I've been surprised, because I thought there'd be a very dramatic effect for CP55, but not much effect for THC. And there's actually been more of an effect for THC than I expected. So, but definitely that's what one would predict, is that for Win and for CP55, that you'd see a dramatic effect, but less so for THC. So, yep.
Okay, in the interest of time, we'll say thank you to Daniel for a very nice talk, and we'll move on to the next talk. Thank you, Daniel. So I believe our next talk is from Helen Collins. Hi. So Helen is going to tell us about immunohistochemical evidence that paroxetine discontinuation in mice is associated with increased activation of 5-HT neurons.
Yeah.
So please take it away.
Great, thank you. So yeah, I'm Helen. I'm a final year DPhil student at the University of Oxford. And yeah, I'm going to be talking about immunohistochemistry experiments that look at paroxetine discontinuation, and specifically the evidence that we have that this is associated with increased activation of 5-HT neurons. So it's been known for several decades now that the abrupt cessation and even tapering of SSRI antidepressants can produce a really debilitating discontinuation syndrome in patients that's highlighted here in a recent review from the Lancet Psychiatry, and includes symptoms such as kind of general somatic symptoms, dizziness, vertigo, there's sleep disruption, GI symptoms, cognitive symptoms, and particularly for my lab, affective symptoms. Anxiety is a particular problem in patients. And so these symptoms appear within 1 to 7 days after treatment cessation, and although they're generally thought to resolve within a couple of weeks, there is evidence that they can last for many months in some patients. Moreover, a recent parliamentary report found that up to half of people coming off their SSRI will experience these symptoms, making it a much bigger clinical problem than had previously been recognised. It's important to note that SSRI discontinuation syndrome is distinct from relapse of depression, as the symptoms appear much faster and include the emergence of these somatic symptoms. It's also interesting that discontinuation syndrome is particularly problematic after discontinuation from SSRIs with shorter half-lives. So paroxetine has the shortest half-life in patients and is particularly problematic. So you might have seen discontinuation syndrome has been in the news a lot recently, and although we've now got a pretty good idea of the clinical situation, there have been very few preclinical studies on the condition. So I've picked out just a couple of papers here, the first on the left from Bosker and colleagues from 2010 that reported increased anxiety-like behaviour following citalopram discontinuation, and an even older study from 1993 that showed ongoing changes to 5-HT metabolism following fluoxetine discontinuation. But other than these, there really has been very little investigation of the 5-HT system, or indeed any part of the brain, during the discontinuation period. And so we recently reported that we'd found increased anxiety-like behaviours in mice 2 days after paroxetine discontinuation. And we've kind of been using a range of different techniques to probe the 5-HT system during the discontinuation period. But it's interesting to note that increased anxiety, like we see in our discontinuation mice, is often associated with 5-HT neuron activation. And so the aim of this study was to basically determine the effect of paroxetine discontinuation on the expression of an activity-dependent gene, CFOS. This can be used as a marker of neuronal activation in 5-HT neurons. So the first thing we wanted to do was to confirm that our mice undergoing paroxetine discontinuation do have increased anxiety-like behavior, and also to assess the time course of this effect. So in these experiments, I've got 3 groups. I've got a saline control group that received saline injections throughout, a continuation group that received paroxetine injections throughout, and then a discontinuation group. These mice received 12 days of paroxetine injections, and then I switched them to saline to mimic drug discontinuation. And in 2 separate cohorts of mice, I either test them on discontinuation day 2 or discontinuation day 5 on the elevated plus maze. So the EPM is a really common rodent test of anxiety-like behavior, and we would expect for a more anxious mouse to prefer being in the closed arms. It's going to spend less time in the open arms, make fewer entries, and have an increased latency to first enter the open arms. So after I've tested the mice on the EPM, I then perfuse them and perform immunohistochemical studies, fluorescence immunohistochemistry, in the dorsal raphe nucleus. So this is the key 5-HT synthesizing region of the brain, and is also the part of the brain that sends serotonergic projections to the forebrain. And so the image on the right is an example of these images where I've stained for CFOS. This is a transcription factor that is expressed in neurons that have been activated, and that's in green. TPH2, which is the synthesizing region, enzyme for 5-HT, so that's marking 5-HT neurons, and NUN, which is a general marker for neuronal nuclei. And in these experiments, I've quantified 3 different kinds of cells. The first, marked by arrow number 1, is a CFOS positive, TPH2 positive neuron. So this is a 5-HT neurons that's expressing CFOS. It's been activated by the behavior. The second type, marked with number 2, is CFOS positive but TPH2 negative neuron. So it's a different kind of neuron in the raphe. We can't say exactly what type it is, but it's not 5-HT. And finally, we also quantified the total number of TPH2 positive cells to check that our treatments weren't affecting the total number of 5-HT neurons, that they're specifically kind of activating cells. And so firstly, we looked at anxiety-like behavior, and we saw that paroxetine discontinuation transiently increased anxiety-like behavior in mice. So on discontinuation day 2, our mice undergoing discontinuation, this dark blue bar here, spent less time in the open arms, they made fewer entries, and had an increased latency to first enter the open arms, all of which is indicative of increased anxiety-like behavior. Interestingly, we also saw reduced distance traveled on the EPM in these mice. And so just to check that this wasn't some kind of general locomotor issue, we also tested them in a neutral environment and saw no general locomotor deficit, suggesting that this is kind of evidence that the mice are more anxious. It's behavioral inhibition in the anxiogenic EPM, so further evidence of increased anxiety-like behavior. So we saw this effect on discontinuation day 2. We then went and looked at discontinuation day 5, but we didn't see any effect on anxiety-like behavior. The discontinuation mice very much resembled saline controls. So overall, this suggests that although we've got this acute anxiogenic effect on discontinuation day 2, it doesn't seem to persist to day 5. It looks like quite a transient effect. So we then went on to look at what was happening to 5-HT neuron activation during the discontinuation period, and we saw that 2 days of paroxetine discontinuation increased CFOS expression in 5-HT neurons. So starting on the left, we looked at total CFOS in the dorsal raphe and saw that it was reduced by chronic SSRI treatment and returned to saline control levels following discontinuation. But when we looked specifically at different kinds of neurons, we saw that in our CFOS positive, TPH2 positive neurons, these are our 5-HT neurons, CFOS expression was increased, suggesting that discontinuation is associated with increased 5-HT neuron activation. And this was specific to 5-HT neurons, as when we looked in CFOS positive, TPH2 negative neurons, there was no effect of discontinuation. And we also checked there was no kind of change in overall number of 5-HT neurons. So this is a specific activation of 5-HT neurons. So because we saw the increased anxiety-like behavior and this increased activation, I correlated these 2 things. So increased CFOS expression in TPH2 neurons was negatively correlated with the time the mice spent in the open arms of the EPM, suggesting that increased CFOS expression in 5-HT neurons is predictive of increased anxiety-like behavior. So we saw this really nice kind of correlation on discontinuation day 2, and because this effect had dissipated by day 5, we fully predicted that the effect on the 5-HT neurons would also have gone away. However, when we looked on discontinuation day 5, CFOS expression was still increased in the 5-HT neurons. So again, looking at the whole RAF-A, in this case, the number of CFOS-positive neurons was increased following discontinuation overall. It was also specifically increased in our TPH2-positive neurons, so again, 5-HT neuron activation. And although there was a slight trend in the TPH2-negative neurons, overall there was no effect. So despite the fact that we don't see behavior at this time point, we're seeing increased activation of 5-HT neurons. And just to confirm this, I correlated the two, and there was no correlation between CFOS and TPH2 neurons and anxiety-like behavior at this point. So just to summarize, 2 days of paroxetine discontinuation is associated with increased anxiety-like behavior in mice, but this effect had dissipated after 5 days. Importantly, though, paroxetine discontinuation at both 2 and 5 days was associated with increased expression of CFOS, which was co-localized with TPH2 neurons, suggesting there was increased 5-HT neuron activation. Moreover, the percentage of CFOS-positive TPH2-positive neurons was associated with increased anxiety-like measures on day 2, but not on day 5 of discontinuation. And so overall, the current data suggest a persistent increase in the activation of 5-HT neurons following paroxetine discontinuation, although this effect only partially tracked with measures of anxiety. And so what we think might be going on kind of generally in discontinuation is that you get a chronic suppression of the system by chronic SSRI treatment that then potentially rebounds above baseline. You get this rebound activation of 5-HT transmission, potentially linked to the symptoms. And we're currently doing experiments to investigate the causal link between this increased 5-HT neuron activation and anxiety-like behavior. So I just want to thank my co-authors on this study, my supervisors, Trevor Sharp, David Bannerman, and Raquel Panaccio, and Caroline, who is an MSc student who worked with us on this project, and the Wellcome Trust for funding this research. So thank you very much for listening, and I look forward to taking any questions.
Thanks, Helen. Really, really nice data. Open for questions? If anyone has any questions, if you want to come up to the microphone.
Very nice presentation. I just wondered, I may have missed it, but did you actually look at your immuno stuff, the CFOS, in naive behavioural mice, i.e. mice that didn't go into the behavioural test?
So we haven't in this case. Obviously, this is an issue. It could be that likely what is happening is that the test is activating CFOS expression. CFOS at baseline is very, very low. It doesn't tend to be expressed, so you do tend to need a trigger. But you're absolutely right. What we can't pick apart from here is what contribution of that increased CFOS is basal activation of the 5-HT neurons and how much is a response to the test. I have a feeling it's an increased response to the test as opposed to increase at baseline, but no, we didn't look at test-naive mice, no.
Yeah, I had a question about the dosing regime that you picked. So you 10 mg per kg for 12 days?
For 12 days, yeah.
And how does that relate to clinical exposure in a human population? And can you just correlate you've obviously seen this nice discontinuation 2 to 5 days. How do you think that relates to clinical use of the drug?
So it's obviously substantially shorter than what we would expect to give to patients. And I think that's something that, translating between mouse and human studies, the timescales are very different. A chronic experiment in mice is 28 days, and we can debate whether that makes it a good model or not. We picked 12 days because 2 weeks is a pretty standard treatment. We did try 28 days of treatment and saw very similar effects after we took the mice off. We did it after just 7 days and didn't see any effects. So we think the 12 days was long enough to see effects, but not so much that it became a very, very long experiment for me. In terms of the duration of the effects, obviously in patients, the reason we started doing this project is because patients were experiencing symptoms for potentially months at a time. And obviously this part of the syndrome, we're only seeing very acute anxiogenic-like effects. We've not been able to go through and do a kind of systematic, what are the different effects on other behaviors. So maybe we're missing something. The fact that there's ongoing changes to 5-HT throughout the 5 days that we see suggests there's more going on. But I think there's probably, I'm sure it gets more complicated. The longer you're on treatment, the more kind of neuroadaptive effects you get. So I think while we're picking up effects, it's far from a full model of the syndrome. We just wanted a way in to start looking at 5-HT, basically.
Okay, thanks. Any more questions for Helen? If not, we'll say thank you very much.
Great, thank you.
Nice talk, thanks. colourful. Okay, our third speaker is Maria Roznovcova, and she's going to talk on the MIDAS motif of the voltage-gated calcium channel CASH-D1 subunit and its contribution to protein trafficking.
Hello everyone, and thank you Gary for the introduction. My name is Maria. I'm a second-year PhD student at the University of Reading, and my project is sponsored by LifeArc. So today I'm going to tell you about the role of the MIDAS motif in the expression and trafficking of the voltage-gated calcium channel subunit CASH-D1. So let's have a look at some background. So what is CASH-D1? CASH-D1 stands for calcium channel and CASH domain containing protein 1. It is encoded by the CASH-D1 gene, which is located on chromosome 1. And the resulting protein is a type 1 transmembrane protein. Studies in mice show that the CASH-D1 is widely expressed in the mammalian central nervous system, including the thalamus, hippocampus, dorsal root ganglia, and cerebellum, which shows similar expression pattern to T-type calcium channels. So why do we study CASH-D1? CASH-D1 was recently identified as a modulator of T-type calcium channels, which are known to be associated with neuronal excitability and play a role in pain signaling. The activity of these channels is increased in chronic pain conditions, including traumatic nerve injury or chemotherapy-induced neuropathies and many more. So this makes the channels possible drug targets to treat chronic pain. However, there have been issues with previous clinical drugs, including ethosuximide, which lacked selectivity for T-type calcium channels and had side effects, and mibefrodil, which had to be withdrawn due to serious drug interaction issues. Therefore, if we can't target the calcium channels themselves, we could target the proteins that interact with these channels. This strategy is already in use for other calcium channels, where the auxiliary subunit alpha to delta is the target of gabapentin and pregabalin that are already used to treat pain. However, T-type calcium channels do not utilize alpha to delta subunits as their modulators, and therefore another modulator such as CASH-D1 could be targeted instead. CASH-D1 is also an alpha to delta-like protein, showing structural homology to alpha to delta subunits. For example, they share an N-terminal signal sequence, von Willebrand factor A domain, and CASH domains. As you can see, alpha to delta has 4 CASH domains. However, the most recent paper suggests that it could actually be 2 double domains, where CASH1 and CASH2 would form first double domain with the von Willebrand factor A domain in between, and CASH3 and CASH4 would form the second double domain. And as you can see here, CASH-D1 only has 2 CASH domains. There are also many differences. For example, alpha to delta subunits are heavily glycosylated. They have a fully conserved or perfect MIDAS motif. They have a gabapentin binding site where alpha to delta 1 and alpha to delta 2 have the site consisting of 3 arginines, and alpha to delta 3 and 4 have the middle arginine replaced with aspargine. And they are also thought to be GPI anchored. On the other side, CASH-D1 only has 7 glycosylation sites. It has a variant MIDAS motif, a variant gabapentin binding motif, which consists of arginine, serine, and another arginine. And it also has a transmembrane domain with a long C-terminal tail. So today I'm going to talk about the MIDAS motif, which stands for metal ion-dependent adhesion site. It is located in the von Willebrand factor A domain, as you can see here, which was shown to be essential for the trafficking of voltage-gated calcium channels. So when it comes to the MIDAS motif, there are differences in the 3 key residues. So CASH-D1 has a variant MIDAS motif, where the 3 key residues are aspartic acid, glycine, and serine. And alpha to delta, in alpha to delta, the glycine is replaced with another serine. And while a fully conserved MIDAS motif is not required for metal ion binding, it is essential for the trafficking and synaptic function of voltage-gated calcium channels. So we utilize a number of techniques to investigate the role of the MIDAS motif. We used PCR mutagenesis to generate two mutants. First mutant, we mutated the glycine to serine, which resulted in the three key residues being identical to those found in alpha to delta subunits. And for second mutant, we mutated the three key residues to alanine, which is a mutation that was previously studied in alpha to delta subunits. We then use Western blot, densitometry, immunohistochemistry, and whole cell patch club electrophysiology to investigate the expression, trafficking, and function of CASD1. So first, we looked at the total protein expression levels of CASD1 in HEK cells. We use Western blot, and as you can see here, the AAA mutant seems to be expressed at a considerably lower level compared to the wild type and the alpha to delta-like G236S mutant. To see if this was a significant decrease, we did densitometry on the Western blots and then analyzed the raw data. On this graph, for normalized data where we consider that CASD1 wild type was expressed at 100%, you can see that the alpha to delta-like G236S mutant was expressed at about 93%, which was not a significant decrease. However, the AAA mutant was expressed at 27%, which was significantly lower than CASD1 wild type or the alpha to delta-like mutant. Next, we looked at the location of these mutants in the same cells. So we labeled cells with rabbit MYC antibody as our CASD1 was tagged with MYC. And then we fixed the cells and labeled them with mouse anti-MYC antibodies. And as you can see here, the wild type was expressed on cell surface, which is indicated by the white arrows, and also inside the cells, which is indicated by the yellow arrows. Then for the alpha to delta-like G236S mutant, we observed the same expression pattern, where we detected it on cell surface and inside the cells. However, for the AAA mutant, we could not detect it on the cell surface, and it only seems to be present inside the cells. So we previously reported that CASD1 modulates CAF3.1, which is a T-type calcium channel. So this is the current for CAF3.1 expressed on its own. Then when co-expressed with alpha to delta 1 subunit, it does not seem much different. And as you can see, these representative traces show that alpha to delta had no significant effect on the current density for CAF3.1. Then, when we co-expressed CAF3.1 with CASD1, the current seems a lot larger. And as you can see here, CASD1 significantly increased the current density for CAF3.1 T-type calcium channels. So as I previously mentioned, the AAA mutation was previously studied in alpha to delta subunits, especially in alpha to delta 1 and 2. And interestingly, the alpha to delta 1 subunit was affected in a similar way as the CASD1, where it was not trafficked to the cell surface. However, the alpha to delta 2, the AAA mutation had no effect on the trafficking or expression. But when it comes to the effects on currents, the AAA mutation virtually abolished the ability of alpha to delta 1 and 2 to increase any currents for the appropriate channels. So to summarize, the AAA mutation decreased the total amount of protein and reduced the expression of CASD1 at the plasma membrane in stable HEK cells, while the G236S mutation had no significant effect on the total protein expression levels or the trafficking to cell surface. And this suggested that the variant MIDAS motif in CASD1 may function in a similar way as the fully conserved MIDAS motif in alpha to delta subunits. CASD1 also significantly increased the current density for T-type channels, unlike alpha to delta 1. And therefore, it would be interesting to see how the mutants, alpha to the AAA mutant and the G236S mutant, affect the channel function, as we did not have the chance to study this yet. We are in the process of making some stable cell lines, and we'll hopefully have the data soon. So with this, I would like to thank my supervisors, Dr. Graham Cottrell and Professor Garen Stevens, my industrial sponsor, LifeArc, and our collaborator from University of Virginia, Dr. Manoj Patel. And thank you all for listening.
Thank you, Maria. Very nice talk. Of course, I have to declare a conflict of interest in that Maria is one of Maria's supervisors, so I won't ask her any questions. Not now, anyway. So has anybody got any questions for Maria? If you have, please come up to the microphone. Last chance, anybody?
Might as well chip in here.
Please. Can you go to the microphone just so that everybody can hear you? Thank you.
You mentioned gabapentin binding. It's a bit out of my field. Does CASH-D1 bind gabapentin, or does gabapentin bind CASH-D1? Did I get that?
I'm in the process of actually looking at this, so I don't actually know yet.
So
But
the binding site for gabapentin,
So it's a variant binding
alpha-2
site.
delta.
So it has a different binding site than the one in alpha to delta 1 and 2. So the variant binding site in alpha to delta 3 and 4 do not bind gabapentin. So we
Okay.
don't know for CASH-D1 yet. We'll have to see.
Thank you.
Another question coming up. Thank you.
Oh, sorry. Yeah, quick question. So very nice data. Thank you very much. Lovely talk. So CASH-D1 seems to increase the size of your T-type calcium channel currents. Is that because you've got more calcium channel protein in the membrane? Or are you changing some of the biophysical properties, such as the nature of the inherent inactivation, be it calcium dependent or calcium independent inactivation? That's my first question.
Okay, so to answer your first question, I believe this is because CASH-D1 actually modulates the currents of T-type channels. I'm going to be looking at whether the CASH-D1 increases the expression of cell surface of the T-type channels a bit later on. So we'll see if that's that, but I believe that just modulates the currents for now.
But you didn't do anything like an inactivation protocol or anything like that to look at potential inactivation states?
I believe there was an inactivation protocol.
Yeah, that was in the paper that we published before Maria arrived. So it does change the open probability of the channel. So there's biophysical effects, not subtle effects on activation and inactivation gating.
Okay. So my second question then is, does CASH-D1, I can't even say it, you're studying it against T-type calcium channels. Does it interact with any other ion channels?
So there was a report that showed that CASH-D1 interacts with CAF2.1, and it can also compete with alpha to delta for the interaction with CAF2.1.
So that's another calcium channel. What about other ion channels?
Oh, that we don't know.
You don't know. Okay.
I don't think anyone looked at that yet.
Okay. And then my final question, and it is my final question, what do you think the stoichiometry is, i.e., how many CASH-D1s interact with the pore-forming ion channel protein? Is it 1-to-1 or 2-to-1?
To be honest, I have no idea.
You don't know. Okay, sorry. Naughty questions. Thank you very much. Lovely presentation.
Thank you.
Thank you for those questions. Ian, I think it was Ian. I can't see it. It's a bit dark here. Thank you, Ian. Okay. If there's no more final questions, we'll say thank you to Maria for a very nice talk. Okay, for our fourth talk, I'd like to invite Chris Sobey. And Chris is going to tell us about reduced long-term motor impairment after delayed systemic amniotic epithelial cell therapy in a mouse model of chronic stroke. I hope I got that right.
Thanks very much, Gary. So as we all know, ischemic stroke is an enormous problem. It's caused by the occlusion of a cerebral artery. It's the second leading cause of death, leading cause of permanent disability, and there are millions of first strokes and stroke-related deaths every year. But we still only have one drug-like therapy that can be used in acute stroke patients. This is the clot-buster drug, tissue recombinant plasminogen activator, so the clot-buster drug. And because of the time restraints in identifying that or confirming that the stroke was in fact a blockage, and the requirements for scanning. There's only about 15% of patients who are able to even receive this therapy. So we've been very interested in looking for alternative ways to treat initially acute stroke with different therapies. And one therapy that we've been very interested in is these amnion epithelial cells. The amnion, of course, is the sac that a human baby grows in. The epithelial cells line the internal surface of that amnion. It's been long known to have healing properties. These cells can be harvested. The tissue is discarded after birth, but the tissue can be, the cells can be collected and stored, frozen down in a GMP manner, thawed and injected intravenously, and they have a lot of properties that are potentially useful in that post-stroke brain injury scenario. In particular, they lack telomerase, so that they don't form tumours. They have high levels of expression of HLA-G, and so they essentially go under the radar of the immune system of the recipient. And so there's no requirement for immunosuppressant drugs. But in particular, they have very strong immunomodulatory properties. So they release many different molecules that, in general, tend to coordinate the inflammatory environment, generally make it anti-inflammatory and promote healing. We, about 4 years ago now, published a lot of work that we did looking at these cells injected intravenously in mostly mice, but also marmosets after acute stroke, within 90 minutes of ischemic stroke. And we found a strong effect of reducing infarct development in these animals, and it's associated with things like much reduced brain inflammation, fewer leukocytes coming into the brain. This work has been translated into a phase 1 clinical trial and soon to be a phase 2 clinical trial in acute stroke patients. We're now exploring what the sort of therapeutic time window might be for usefulness of these cells beyond that initial period of a few hours. So the aim of this study, these ongoing studies that I'm showing today, is to test if administration of these human amnion epithelial cells between 1 day and 5 weeks after stroke might promote long-term functional recovery in mice. Most of the work is looking at motor function, but we also have some data on cognition. So the model of stroke we used is the photothrombotic model of cortical stroke. We've used aged male and female mice, so 15 to 18 months of age. With this model, rose Bengal dye is injected intraperitoneally, allowed to circulate for a few minutes, and then after that, cold light can be directed transcranially to the part of the cortex that we want to target a stroke in. And then we're given mice either vehicle or 1 million cells intravenously post-stroke in different cohorts, either at 1 day after this stroke, at 7 days after the stroke, or 14 and 35 days, or 2 weeks and 5 weeks after stroke. And we perform functional testing with a cylinder test along the way. So a bit more about the model of stroke. I mentioned rose Bengals injected. The light can be directed, targeted to the zone of interest. So, for example, the primary motor cortex on the left side will manifest as impairment of the right forepaw in a cylinder test. The prefrontal cortex will result in cognitive impairment, and this is what we're looking at. Over the longer term, it's a relatively small stroke compared to the middle cerebral artery occlusion model, and therefore it's much more suitable for long-term studies. The cylinder test, of course, measures motor impairment. The readout is asymmetry of four-pore preference for timing against the side of the cylinder. In general, we do a baseline test and repeat the test over that 8-week time frame. So we looked at males, females, and then combined the data. Here's the data from males injected 1 day after the stroke. The baseline data was prior to the injection, and at 1 week, there's a separation between animals that receive vehicle and cells, and this has reached statistical significance at week 8. Females are generally the same sort of profile. In these cohorts, there's probably a little bit of a faster response or a larger difference at 1 week. And combining the data shows a fairly sustained improvement in motor impairment over 8 weeks when the cells were injected at 1 day. When the cells were injected at 7 days, we actually see no real effect in males. This is week 1. This is just prior to the injection, and at week 5 and week 8, there's no difference really between vehicle and cell-treated animals. In females, it looks a bit different, and there seems to be a statistically significant effect, a bit of variability there, but overall there looks to be some benefit in females here when the cells are injected a whole 7 days after the stroke. In combination, there's sort of borderline, I think that was 0.06 p-value, but clearly that effect, any effect, is coming from females here. Finally, with the motor impairment, there's basically no effect when we inject cells as late as 2 weeks, and we top them up again at 5 weeks in males or females. And there's the combined data. So I just want to finish briefly with one slide of data where we've looked at the cognitive impairment that we can produce by the prefrontal motor cortex stroke. We've used a Barnes-Maze test 4 weeks after the stroke with daily tests. The Barnes-Maze test uses aversive stimuli, bright lights, loud noise, which stop when a mouse enters one of 20 holes, or the target hole of 20 spaces. Reference markers are around the room, whoops, and the hole is switched on day 5, so that enables us to look at cognitive flexibility or relearning. But overall, we're looking at spatial working memory, short-term memory, executive function, decision-making, problem-solving. So the data here looks at 4 groups: animals, sham control animals, sham animals that receive cells, stroke animals that receive vehicle or cells 1 day after the stroke. And here we are 4 weeks later looking at the Barnes-Maze test. And you can see that animals that had a stroke but did not receive cells were much slower at the start to find the hole in terms of primary latency. And then the hole was switched from day 4 to day 5, and it took a long time to figure that out. So, but importantly, animals that were treated with cells were much better, kind of similar to the sham-operated animals, and they weren't really confused so much with the switch of the hole. If we look at overall at the overall time taken to not only find the hole but enter the hole, escape latency, really getting at the decision making, you can see again the profile very clear worsening or worse performance of animals that had a stroke but no cells, whereas animals that had stroke and cells were pretty good. So in conclusion, our data suggests that administration of human amnion epithelial cells intravenously at 1 day post-stroke is effective in reducing long-term motor and cognitive deficits after ischemic stroke in aged mice. At 7 days, there may be some effect in females, but at 2 and 5 weeks post-stroke, it appears that it's too late for an effective result with this cell therapy in this model. Just want to acknowledge the work of the people in our group that have done these experiments and our collaborators who have developed this cell therapy. Thank you very much.
Thanks, Chris. The paper's open for questions. Again, if you've got questions, please come up to the microphone. Yeah, at the back.
Hi, Chris, thanks for that. I was wondering, do you think the mechanism behind the beneficial effects here are improved vascular recovery in the brain, or do you think it's something else that the cells are specifically doing?
Yeah, so the concept that we're kind of thinking about is overall sort of reducing the amount of secondary inflammation-driven injury that follows the stroke. But we also have, so all this data, of course, is in vivo, and we're sort of starting to look systematically at the tissue. We do have evidence that there's a thinner glial scar and possibly higher levels of vascularization or post-stroke angiogenesis in that affected area. So you may be right, there could be better blood flow, but it's probably a combination of things.
And so in that case, do you think it would be relevant for something other than stroke to use these cells?
Yeah.
MI or something like that?
In general, we think that any sort of relatively acute, of course this is getting towards the chronic stage, but any sort of acute inflammatory injury where there's, for example, SDF1 generated as a chemoattractant for CXCR4 expressing cells, which these are and most stem cells are, the cells should be able to find their way to the injury and have a beneficial effect. And this membrane, this amniotic membrane, has been known for decades to have sort of pro-healing effects in, for example, burns and eye injuries and so on. So, yeah, certainly not specific for stroke.
Thanks.
Yeah.
Thank you. Number 2, go ahead.
Thanks. Great talk. For the females, did you control for estrus, or were they too old? 15 to 18 months? Yeah,
no, we did not. Yeah, they probably are too old. And of course, because stroke is typically an age-related disease, we try and have most of our studies in these much older animals than historically we might have, and of course it's important to study both sexes as well. We have looked in earlier work in animals, tried to match estrus stage, and we haven't really picked up a clear effect. It's sort of much shorter, of course, in mice, and so you probably have to do a lot of animals to get statistical power to really be sure.
Yeah.
Thanks. Time for one final question at number 1.
Thanks, great work. So the question relates somewhat to the first question, but were you able to follow engraftment of these cells, say in the motor cortex or the prefrontal cortex?
Yeah, we haven't looked at that extensively, but we know the cells get there. We have looked, we have some level of imaging of the cells turning up in the infarcts. What we think happens, though, is that they kind of are effective for a period of days or, maybe a week or two, and then they senesce. So this is not sort of stem cell therapy where we're trying to rebuild brain tissue with cells that are going to generate or regenerate tissue. We think this is really just the cells getting there for a relatively short period of time and influencing the sort of the healing of the environment.
Thank you.
Okay, if we've got no further questions, thank you very much, Chris. Very nice talk. Thanks. Okay, we come to our final presentation in the neuropharmacology session. This is going to be given by Raj Makwana, and he's going to talk about RJ vasopressin, and adrenaline act in synergy.
Nausea or the urge to vomit is associated with the central mediate increase in interoception. Nausea is also associated with dysrhythmic electrical activity of the stomach, which arises from the interstitial cells of Carl located in the stomach wall. These depolarize spontaneously and work with the muscle and enteric nervous system to initiate contractions of the stomach. Other physiological changes associated with nausea include a rise in plasma concentrations of vasopressin and adrenaline. So adrenaline is released from the adrenal glands and vasopressin is released from the pituitary. These have nosogenic activity. Current assumption is that all nosogenic substances act on the area postrema, which is located outside the blood-brain barrier. But the evidence is weak. We hypothesize that the release of adrenaline and vasopressin affects human gastric motility by modulating the excitability of the interstitial cells of Cajal. Such changes are then detected by the vagus nerve and signaled to the brain for interpretation as nausea. The aim of my work was to investigate the effects, interactions, and site of actions of these two hormones on human stomach contractility in the presence of tetrodotoxin to block neurotransmission. Human stomach was obtained following informed consent from patients undergoing a vertical sleeve gastrectomy. A 5 centimeter square section from the top and bottom end of the gastric sleeve was cut. I will refer to this as the proximal and distal stomach. The muscle was dissected away from the mucosa and used for 2 types of experiments. Firstly, to measure muscle contractions and examine the actions of adrenaline and vasopressin. These involved looking at muscle contraction, circular muscle, in using standard tissue bath methods. The probes contain tetrodotoxin to block neural activity by blocking sodium channels. Secondly, to localize the receptors on the pacemaker cells, of the interstitial cells, Cajal, and the smooth muscle. So these used antibodies directed towards c-KIT, which is a selective marker of the interstitial cells, Cajal, and alpha smooth muscle actin, which is a marker of smooth muscle. This slide shows the actions, this slide shows the contractions of the proximal and distal stomach in the absence and presence of increasing concentrations of vasopressin applied cumulatively. So in the absence, we can see that the muscle is spontaneously active. There are smaller contractions of the proximal stomach compared to the larger contractions of the distal stomach. Vasopressin causes an increase in muscle tone, so there's an increase in baseline. It increases the amplitude of the contractions relative to baseline levels and also increases the frequency of the contractions. This data can be presented as log concentration response curve. So the x-axis represents the concentrations of vasopressin. The top panel shows the changes in muscle tone. So we have the green line, which shows the control responses. And then in the presence of increasing concentrations of an antagonist of the vasopressin V1A receptor. So the middle panel shows changes in the amplitude caused by vasopressin in the presence and absence of the V1A antagonist, and changes in the frequency. The muscle tone has been expressed relative to carbachol, which is a muscarinic agonist applied at the end of the experiment to estimate maximum contractility. Changes in the amplitude and the frequency of contractions are expressed relative to baseline values. So the rightward shifts caused by the V1A receptor antagonists are parallel without reduction in the maximum effect. Shield analysis generates shield slopes not different from immunity and with PKB values of 9.5. And this is consistent with the V1A receptor activity. So vasopressin acts on V1A receptors. This slide shows the actions of adrenaline, and the action is slightly complicated. At low concentrations, we see an increase in muscle tone, and at high concentrations, there is relaxation. Similarly, at low concentrations, we see increase in the amplitude of the contractions and also frequency. So this is similar to what we see in the distal stomach. This data is presented as low concentration response curves for the three changes in this is the muscle tone, amplitude, and frequency of contractions. Over here, the green line shows the controlled responses. The blue line shows the actions of adrenaline in the presence of an alpha-1 receptor antagonist of the adrenaline receptors. And the red line shows the actions of adrenaline in the presence of propranolol, which is a beta-2, which is a beta adrenaline receptor antagonist. So if we focus on the top panel, what we see is that the changes in muscle tone, so the increase in muscle tone is antagonized by paroxetine, whereas the relaxation is blocked by propranolol. And PA2 values for paroxetine confirm that adrenaline is acting on the alpha-1 adrenaline receptor. And the inhibitory effects are blocked by propranolol. So adrenaline excites via the alpha-1 receptors and inhibits through beta receptors. Now, there are clear notable differences between the spontaneous activity of the proximal and distal stomach. You will notice that there is a difference in the amplitude of the contractions. Distal stomach contractions are almost 5 times larger. The other key difference is the rate of contraction development and decay. This is similar in the proximal stomach, whereas in distal stomach, the contractions develop much faster and decay slowly. We've previously shown that these spontaneous contractions are unaffected by blockade of sodium channels with tetrodotoxin, but they are reduced by inhibition or blockade of the calcium-dependent chloride channel, anoctamin A1. So this is a blocker of the anoctamin 1 receptor. So this is suggesting that the spontaneous contractions are not neuronal, but rather they are myogenic. So we wanted to know how vasopressin and adrenaline alters the spontaneous contraction waveform. So we developed software to analyze various features of the contraction individually. So we looked at changes in the frequency of contractions before and after an agonist was applied. We looked at changes in muscle tone. For each specific contraction, before and after an agonist, we measured the amplitude, the rates of contraction development, decay, changes in the area under the curve, and the duration. So this is a link to the software which we used. Using the software, we can extract these quantitative values automatically and then use other commercially available software to plot a radar plot. So each arm corresponds to the different features of the contraction waveform. So we have tone, amplitude, and frequency. So what we see is that the frequency of contractions of the proximal distal stomach are similar. These are similar to electrical slow wave activity in vivo of around 3 per minute. So the data over here is expressed as mean plus standard error of the mean. And the asterisk corresponds to the significant differences using t-test. So we looked at what adrenaline and vasopressin does at sub-maximum concentrations. So these were concentrations that elicited 75% of the maximal effect. And what we find is that vasopressin and adrenaline increase the amplitude of the contractions compared to baseline levels, but when applied together, their effect is greater than the sum of their individual effects. So this was a synergistic action. This also happens in the distal stomach. So we get a much more exaggerated response when both hormones are applied together, and this is greater than the sum of their individual effects. We then looked at where these receptors were expressed. So we used antibodies and stained the muscle and interstitial cells CAHA. So on the top panel, c-KIT is a positive, is a marker of the ICCs. The blue stain corresponds to DAPI, which is the nuclear counter stain. c-KIT stains the projections which are of the ICCs. The red stain is for vasopressin, and when we look at the merged image, we see that the vasopressin V1A receptor is located on the c-KIT positive ICCs. Similarly, the alpha-1 adrenoreceptor is also expressed on the c-KIT positive ICCs. We also find that both vasopressin and alpha-1 adrenoreceptors are also co-localized with this alpha-smooth muscle actin, so they're found on smooth muscle. So these graphs show that all smooth muscles express the V1A receptor, and only around 40% of the smooth muscle cells express the alpha-1 receptor. In contrast, both V1A and alpha-1 receptors are expressed in similar levels, or of around 60% on ICCs. So in conclusion, there are clear differences between the spontaneous contractility of the proximal distal stomach, which probably reflect the physiological functions, where the proximal stomach is involved in accommodation, it stores food, and the distal stomach is involved in digestion. So powerful contractions are needed to break food down. Adrenaline and vasopressin act through the specific receptors. So adrenaline acts on the alpha-1 adrenoreceptors, vasopressin acts on the V1A receptors. These receptors are located on both the interstitial cells of CAHA and the muscle. And they stimulate these cells at concentrations found in people experiencing nausea. So together, they act synergistically at low concentrations to elicit much more profound increases in gastric motility, which would then be signaled to the brain for interpretation as nausea. So the take-home message is that both hormones can act synergistically to excite gastric motility by acting on the ICCs and muscle, and this will lead to nausea, and these effects are consistent with their nausogenic activity. I would like to end by acknowledging the fund sources and our colleagues who are involved in discussions, technical assistance, and administrative help. And I would also like to add Dr. John Broad is over here to this list, who was involved in the exploratory experiments. Thank you.
Thanks, Raj. Really nice talk. Do we have any questions for Raj?
Very nice talk indeed. So, what are you waiting for? Are you in the clinic?
So if you could just bear with me for a minute, I'm hard of hearing, so...
oh, sorry. So,
Danny, Raj has asked that I write the question down, and coincidentally, your question was the same as mine, so I've already written it down.
Okay, sorry.
So yeah, I guess the general question was, what is the clinical relevance of this?
Well, it seems that when two hormones, which otherwise have very low efficacy, when they act together, produce very profound changes. So, firstly, the whole basis of nausea is very different because the types of different nausea. We might have nausea induced by motion sickness, maybe nausea due to pregnancy. So, depending on the different types of hormones, you might get different types of nausea, and of course, blocking their individual activities may not be an ideal strategy for developing anti-nausogenic actions. So, in terms of clinical, well, it's difficult because we don't know the full mechanism of how nausea arises and how do we block it.
Okay. Do we have any further questions? We're actually right up against time, and I know we've been told to finish these sessions by 6 o'clock. So would you say thank you to Raj for a great talk? And thank you to all the other speakers in the session. I hope that everybody enjoyed the session.
Thanks.
Thanks, Raj. Yeah, so with that, we'll just maybe a final round of applause for our speakers. Yeah, and enjoy the rest of the meeting. Thanks.